Next up is oral presentation number 28, Toronto, Ontario, Canada, University of Toronto Schools. It's a Mission 8 finalist proposal with 11th and 8th grade student researchers.
Good afternoon, ladies and gentlemen. Today, we will be discussing our experimental proposal for Mission 8 of SSEP. We are from University of Toronto Schools, also known as UTS, from Toronto, Ontario, Canada. The main question that we aim to explore is, how will microgravity affect the functions and development of stem cells in the Mediterranea, a Planarian flatworm? My name is Rahim, and I, along with my fellow researchers, Arjun, Maddie, Matthew, and Alicia, will be talking to you today about sending Planarians to space. Now, some of you may be thinking, what are Planarians? Planarians, as shown in this image, are non-poisonous flatworms about 20 millimeters long that live in aquatic ecosystems. Planarians have the ability to regenerate large parts of their body with pluripotent and totipotent stem cells. In addition, many planarians reproduce asexually, a process in which planarians divide in half when there is a low density of planaria in their habitat. When planarians are deprived of food, they proportionally decrease in size and mass without affecting their natural processes. Furthermore, this process allows planarians to survive for extended periods of time without food. They also have very similar nervous and physiological systems to that of a human, and making them, therefore, very important to research. Now Arjun will be discussing stem cells.
Stem cells are unspecialized cells which are able to differentiate into one or more types of cells. There are two main types of stem cells, namely embryonic and adult stem cells. The diagram shown here displays totipotent, pluripotent, multipotent, and unipotent stem cells, each of which have a varying ability to develop into different types of tissue. Our experiment in particular will focus on totipotent and pluripotent stem cells. When stem cells undergo mitosis, they split into two different cells, one of which can further divide as a pluripotent stem cell, and one that can no longer divide, referred to as daughter stem cells. Next slide. In planaria, the daughter cells are dispersed throughout the outer edges of the planarian, while the adult stem cells remain in the center, as you can see in this picture here. If any part of the planarian is injured, the stem cells assemble at the location of the injury and initiate a healing process. In other words, the arrangement of the stem cells allows for regeneration on a mass scale. As you can see in this next image here, if a planarian is cut into 3 separate pieces, each is able to form a whole organism within the short span of just 7 days.
Going to this project, we're asking 3 different, however related, questions. Each targets the investigation of how microgravity affects stem cells from a different angle. Our first question, as you can see here, how will microgravity affect the differentiation of embryonic stem cells in a planarian egg? Our second question was, how will exposure to microgravity affect Mediterranea stem cell functions such as regeneration on their return to Earth? Our third question was, how will a microgravity environment affect the differentiation processes of adult stem cells? Okay. Here can be seen the layout of our fluid mixing enclosure. Each clamped off volume holds a separate experiment, each geared towards investigating one of our 3 research questions as outlined on the previous slide. With this design, we are making the most efficient use of a limited volume. The first volume of our FME will hold eggs of the sexual strain of the S. Mediterranea. The differentiation process that occurs in these eggs is different from the regenerative differentiation in the other 2 experiments in that it parallels a human specimen much more closely. This is because zygotic stem cells in most organisms, humans included, are totipotent as well, like the S. Mediterranea.
In the second volume of the FME will be 15 photo-bleached S. Mediterranea worms. Essentially, this photo-bleaching process harmlessly removes the pigment from the tissue of the organism. One of the natural stem cell-dependent processes of these planaria is constant regeneration of tissue. And because this new tissue will be pigmented, we will be able to compare this to the rest of the organism after its time in microgravity. These worms are also very valuable to the experiment because they can be kept alive for use in further experimentation on return to Earth. When they return, we'll be able to see how exposure to microgravity will affect processes such as regeneration, asexual reproduction, among others on Earth. In the third volume of the FME, will contain worms exposed to the chemical solution bromodioxyuridine, or BRDU. And what this substance does is it allows us to label all newly generated daughter cells throughout the organism's time in microgravity. And unlike simply comparing tissue pigmentation, as in volume 2, on their return to Earth, we'll expose the worms to a fluorescent dye, allowing us to view each individual new cell, allowing us to analyze daughter cell distribution, population, and other topics. The disadvantage being that this process requires us to kill the organism to analyze it, preventing its use in further experimentation.
Now, some of you may be wondering why sending planarians to space is so important. Planarians are actually very similar to humans, even though they are incredibly simplistic. More than half of the genes in planarians have parallels to human genes, and in particular, the gene for stem cell regeneration. This means that by sending planarians to space, we learn more about their genes and subsequently our own. We can study whether microgravity increases stem cell regeneration and enhances the newly formed cells. This in turn yields huge benefits for humans. If we find that microgravity enhances stem cell regeneration in planarians, we can produce tissues and cells lacking in humans. For example, individuals with type 1 diabetes have malfunctioning insulin cells, meaning that their body cells cannot uptake sugar. These stem cells can then produce the insulin cells in microgravity. We thank you for listening to our proposal and welcome any questions you may have. Thank you.
That was very good. 1 second to spare. I didn't want to interrupt you, but that was very good. I wanted to say congratulations to our third Canadian team. I also would be remiss to point out that this is one of 3 finalist proposals that the Step 2 National Review Board reviewed here at the Smithsonian's National Air and Space Museum in May. And the subpanel reviewing the UTS schools was just blown away by the caliber of your proposals. So a heartfelt congratulations to all of you for the work that you've done, all 3 teams. I know that Suzanne Monier did an excellent job in coordinating this effort in your community. Let me point out that the North Charleston, South Carolina team that's working on Tin Whiskers was a finalist 2 years ago. And they took the review panel comments to heart and resubmitted the following year, and they were selected as the flight experiment. And that's how real research works. So if UTS participates next year, I would urge you all to consider looking at the review panel comments and resubmit this proposal, because all 3 proposals were exceptional.
Thank you.
Any questions for this team?
Hi, Suzanne from Toronto. I have a question just about the genome. If your experiment, can that apply to any Planaria mediterranei?
Yes, it can, because each Planaria, or Schmidtea mediterranea in this case, they're identical from a genetic standpoint, and that's because they're an asexual strain, and that means that there's no merging of genes, because meiosis, which takes place in sexual species, does not take place. Yes, and an interesting fact is actually that they're exactly identical to the original Planarian strain, which was found in a fountain in Spain. So they're identical.
Hi, I'm Vicki from Johnson County, Nebraska, and my question was the middle experiment. How long does it take them to get the color back, and how can you tell how fast do they need to get back to you so that you can know that it didn't happen on Earth?
First of all, the tissue regeneration is constant, so it's not like a timed thing. It just happens throughout the entire duration of the experiment. And with regards to the second part of your question, we believe that was the major flaw in our experiment that prevented it from going to space, was that with the quarantine period and all and transportation time, it was very hard to ensure that gravity and its exposure to regular Earth conditions could have played a role in the experiment in that volume.
Thank you.
Tim Livengood with NCSE. So let's see. So when these things are refrigerated, does that stop the activity? I mean, essentially slow it down so much that it's effectively stopped?
It may somewhat stop the regeneration, but the problem is that the planarians won't be able to survive being in that sort of free state for like an extended period of time, and they can only survive in that state for a very short period of time, or then they'll die. So that's not a possibility.
I was curious that if, I mean, right now we have difficulty with being able to refrigerate samples on the station or for the return, but it's possible that that could change in the future. I was wondering if that would be enabling or unhelpful on your experiment.
Well, yeah, essentially we did look into that when thinking about the worms at the start of the experiment, but we talked to people at the lab we were working with in Toronto, the Pearson lab, and they said that there really is no way to halt the regeneration, and it takes like very, very large measures even to slow it, because it's just such a powerful process.
Okay, thank you.